Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine your body's immune system, right? It's like this super vigilant security force, always scanning for trouble. Now, what if certain molecules could just, whip out a sort of molecular invisibility cloak to slip right past those defenses? That's basically what a tiny, tiny tweaked RNA does. And this RNA is at the very heart of our, well, incredibly successful mRNA vaccines.
Exactly. And for years, how it pulled off this disappearing act, well, that's been a real mystery.
It really has.
And here's the thing that I think makes this really worth a deep dive today. It isn't just about vaccines, so that's obviously huge.
Right.
It's about understanding something really fundamental. How our immune system tells us apart from themselves, from non-self, right down at the molecular level.
Oh, okay.
So today we're diving into this groundbreaking paper just published. And it finally pulls back the curtain on this molecular invisibility cloak.
And it sounds like there are some surprises in there, too.
Oh, definitely. Some revelations that might actually change how we think about future medicines.
Okay, great. We've got some excellent source material here, this paper primarily. So our mission today is to really distill the core findings, explore this kind of intricate dance between these RNA modifications and our immune sensors.
Sounds good.
Start with maybe a quick primer. You know how the body usually spots foreign RNA.
Yeah, the basics.
Then zoom right in on the specific molecular changes that let this modified RNA fly under the radar.
The cloak itself.
Exactly. And finally, what exciting new insights this gives us. Future breakthroughs, maybe, and especially that unexpected curveball you mentioned about M1.
Yes, definitely need to cover that.
All right, so let's set the scene. Our innate immune system, that's the first line of defense, right?
Correct.
It uses these specialized sentinels called pattern recognition receptors, PRRs. Think of them like, I don't know, tiny security cameras, very specific ones.
Good analogy.
And among the most important here are the Toll-like receptors, TLRs, specifically TLR7 and TLR8.
Key players.
Yeah, and they're kind of hidden away inside these special cellular compartments.
Yeah.
The lysosomes.
That's right, inside the cell. And their job is basically to look out for fragments of single-stranded RNA. Stuff like you get from viruses or bacteria invading.
Okay, so they're looking for RNA fragments.
But, and this is crucial, they aren't just reacting to any old RNA floating around. That would be chaos.
Right, you attack your own stuff.
Exactly. They're incredibly fine-tuned. They detect very specific RNA degradation products, pieces of RNA that have been chopped up.
Chopped up by what?
By our own cellular enzymes. Think of it like a molecular security shredder, inside the cell.
A shredder? Okay.
Yeah, and enzymes like RNase T2 and these PLD exonucleases, they're the key shredders. They chop up the RNA into very specific pieces.
And these pieces then?
They fit perfectly into two different detection pockets within TLR7 and TLR8, like a key in a lock. And that triggers the immune alarm.
Got it. Shredded RNA pieces fit the lock, alarm goes off.
Basically, yes.
Now, here's where it gets clever, right? Our own self-RNA, it doesn't set off these alarms.
Exactly. It has natural modifications, little chemical tags that act like a password, essentially. Yeah. Don't shred me, I belong here.
And one of the main modifications is pseudouridine, or S.
Yes, pseudouridine. It's the most abundant of these natural modifications in our own RNA.
And then, famously, back in 2005, researchers discovered something huge.
Pivotal moment.
If you just replace the normal RNA building block, uridine, or U, with this pseudouridine in lab-made mRNA, it completely shut down the inflammatory response. Just switch it off.
A massive finding. Absolutely critical for making mRNA vaccines work. Without that tweak, the vaccines would cause a huge immune flare-up just from the RNA itself.
Allowing them to deliver their message quietly.
Precisely. But for nearly 20 years, how? How did Psy achieve this immune evasion? That remained, well, kind of a black box.
Until now, hopefully. This deep dive aims to finally crack that box open.
Let's do it.
Okay, so let's dive into the core findings of this new paper. Starting with that key enzyme, that shredder, RNase T2.
Right.
They use something called quantitative mass spectrometry, which sounds like a super precise molecular scale.
That's a good way to put it, yeah. It lets you see exactly what fragments are being produced.
To see exactly where RNase T2 cuts RNA, what did they find? Was it random?
Not at all. That's the intriguing part. They confirmed RNase T2 is incredibly picky, a real fuss spot.
Okay.
It strongly prefers to cut RNA between specific pairs of letters, the nucleotides, particularly where you have an R that's appearing, like A or G followed by a U, uridine, especially GU sites.
Are you?
Yeah. Now, it can cut other places, but only under very limited conditions. Its main targets are those RU sites.
And they compared to other enzymes.
They did. They showed another enzyme, RNase 1, cuts much more broadly. It's not nearly as picky.
So RNase T2 is the specialist here.
Exactly. Which immediately raises the question. If it's so specific about cutting at U, what happens if you change that U?
Okay. Like swapping it for pseudouridine.
Precisely. But first, they wanted to be sure this pickiness held up in more complex situations.
Right. Because a real mRNA molecule isn't just a simple sequence.
Exactly. So they scaled up. They used a longer, more complex RNA molecule, something much closer to actual mRNA. Did RNase T2 still show its preference?
And did it?
It did. Even in this more complex, messy environment, RNase T2 is still laser-focused. It almost exclusively produced fragments ending in specific ways, mostly those UA and UG dinucleotides again.
Consistent.
Very consistent. Meanwhile, good old RNase 1 just kept chopping away more broadly.
Okay, so that really confirms RNase T2's specific job. It sets the stage perfectly.
It really does. Now you know the target site.
So this is where it gets really interesting, right? We know RNase T2 is picky. It likes cutting at U. So the researchers asked, what happens if you swap that U for pseudouridine? Or for N1-methyl-pseudouridine in 1A. That's the one used in the COVID vaccines.
Yep, the therapeutically relevant one.
Did it, jam the shredder?
Jam the shredder is a great way to put it. They found that both A and M1 dramatically impaired RNase T2's ability to cut the RNA.
Dramatically?
Yeah, like if you replace the U in a GU site with Psi or M1-PI-Psi, RNase T2 just struggles. It can't cut there effectively at all.
Wow. Do they know why? Like structurally?
They do. They used cryo-electron microscopy, which gives you these amazing 3D pictures of molecules.
Right.
And they saw that Psi actually distorts the enzyme's active site, the cutting part. It physically changes the shape so the enzyme can't bind properly and make the cut.
So it physically blocks it.
It physically gets in the way, yeah. And this explained the nearly 90% reduction in cutting they measured. It's a massive effect.
And crucially, did other shredders, other RNases, get blocked too?
Nope. And this is key. Other RNases, like the RNase A family, were completely unaffected by these modifications.
So it's selective.
Highly selective stealth. It's like putting a unique coding on a document that only stops one specific type of shredder, RNase T2, while others can still process it normally, if they could.
That's fascinating. So this directly links the modification, Psi, or M1-PSI, to stopping that very first step of immune recognition getting shredded into detectable pieces by RNase T2.
Exactly. The first layer of the invisibility cloak.
Okay, so if RNase T2, the main shredder for this pathway, can't process the modified RNA properly, what's the downstream effect? What happens to the immune alarm, TLR8? Did they test that in actual human immune cells?
Yes, that's the critical connection to make. Does blocking the shredder actually silence the alarm?
And does it?
Resoundingly, yes. They took primary human monocytes, key immune cells, and exposed them to RNA. Unmodified RNA strongly activated TLR8 as expected.
Right.
But the RNA containing Psi, or M1-PSI, almost nothing. Complete failure to stimulate TLR8.
Wow. Consistent effect.
Very consistent. They saw it across different cell types, and crucially, the effect depended entirely on RNase T2 being present and active. If you block RNase T2 another way, you get the same lack of response.
So part 1 of the invisibility cloak confirmed. You physically stop the RNA from being cut up into the specific pieces that TLR8 recognizes. The shredder is jammed.
Precisely. First line of defense bypassed.
But hang on, the story doesn't end there, does it? Because even if the RNA isn't shredded properly, could the pseudouridine molecule itself, maybe if some gets released, directly trigger TLR8, like act as a direct signal?
That's a really important question they asked.
Yeah.
Could Psi itself be a ligand, a direct activator for TLR8?
Yeah. So they tested it, just throwing the individual building blocks, the nucleosides, at TLR8.
They did. They used high concentrations, and what they found was, well, kind of a curveball. Normal uridine, U strongly activated TLR8. No surprise there, that's known.
Okay.
But pseudouridine, all by itself, completely inactive, didn't trigger TLR8 at all. like a silent bystander.
Okay, so Psi itself is inert. That fits the narrative so far.
It does. But here's the surprise. N1-methyl-pseudouridine, M1 from the vaccines,
Yeah.
it did activate TLR8 directly, similar to normal uridine.
What? But it also blocks RNase T2, right?
Exactly. So M1A is this master of disguise for the shredder, RNase T2.
All right.
But if the M1 molecule itself somehow gets presented directly to a TLR8, it can still trigger the alarm.
That's really counterintuitive. So it hides from the processing, but it's still recognizable on its own.
It seems so. They also did biochemical tests looking at how TLR8 molecules pair up, which is part of activation. Psi alone was very weak, basically background noise. But M1A could get TLR8 to pair up, similar to U.
So M1 is sneaky with the enzyme, but not totally invisible to the receptor itself.
That's a great summary. It suggests M1 retains some ability to signal directly, even while evading the main processing pathway. A subtle, but potentially very important difference from Psi.
Wow. Okay. Mine's slightly blown there. Let's pivot then. What about the other main receptor, TLR7? We know Psi-modified RNA dampens TLR7 responses too. How does the cloak work there? Is it the same RNase T2 story?
It's related, but slightly different, involving more players. For TLR7 to get activated, it needs specific RNA fragments too, but one of the most critical signals is a small molecule called 2'3-cGMP.
Okay, a different kind of key for the TLR7 lock.
Exactly. Think of it as TLR7's specific wake-up call. And the researchers found that both Psi and M1-PSI strongly impair the release of this critical messenger.
How? By blocking RNase T2 again?
Yes, partly by blocking RNase T2, but also by inhibiting those other enzymes involved, the PLD exonucleases. So for TLR7, it's like a double block on the shredding and processing machinery needed to generate that wake-up call.
Okay, so less of the 2'3 cGMP signal gets produced?
Much less.
Yeah.
But there's more. They also showed that even if some Psi-containing RNA fragments were produced, they didn't engage the second binding pocket of TLR7 properly.
Ah, so even if a piece gets through, it doesn't fit the lock right.
Precisely. It's like the key is cut wrong. So for TLR7, it's a double whammy. Less signal generated, and the signal that is generated doesn't work properly.
That's thorough immune evasion.
It really is. And just to tie it all together, they did experiments in mice, confirming that RNase T2 is absolutely crucial for detecting normal, unmodified mRNA in vivo. Taking away RNase T2 mimic the effect of using modified RNA.
Validating the whole mechanism in a living system.
Exactly. It really solidifies RNase T2's central role.
Okay, so let's try and synthesize this. What does this all mean? This deep dive seems to reveal a really sophisticated, two-pronged molecular strategy for how these modified RNAs hide from the immune system.
This is a good way to put it. Two main layers of stealth.
First, the modification Psi and M1-PSI, they significantly mess with the processing. They jam up the shredders, particularly RNase T2, but also PLD for TLR7.
Right. The RNA just isn't cut into the right pieces to be easily detected. Step one, bypassed.
And second, even if some fragments or the molecules themselves get near the receptors.
Yeah, the second safeguard kicks in. Psi itself is a really weak signal for TLR8, and Psi-containing fragments don't engage TLR7 properly.
So multiple checkpoints are failed.
Correct. But the really thought-provoking piece, the new insight here, is that difference between Psi and M1-PSI.
Right, that M1 curveball.
Yeah. While M1 is just as good, maybe even slightly better at evading the shredders, it keeps the ability to directly activate TLR8 if encountered as a single molecule, unlike Psi, which is silent.
That suggests a subtle but really significant difference in how these two modifications interact with our immune system, even though they look so similar.
Absolutely. And that opens up entirely new questions and possibilities.
So this work really pushes us beyond just, knowing that Psi mRNA is less inflammatory. It gives us the why and the how at a deep molecular level.
The structural and mechanistic detail, yeah.
For people developing vaccines or RNA therapeutics, this seems huge. It's like you've suddenly got a dial. You could potentially tune the innate immune sensing.
Exactly. Maybe by blending different levels of Psi and M1-PSI, or maybe designing entirely new modifications based on these principles.
Imagine designing an mRNA therapeutic where you want a little bit of immune activation versus one where you want absolute silence.
That's the potential power this kind of detailed understanding gives you. Precise control.
So what are the big remaining questions or maybe the provocative thoughts to leave our listeners with?
Well, one thing the study mentions is this idea of degraded endosomal clearance. If the modified RNA isn't getting shredded properly by enzymes like RNase T2.
Does it just hang around longer inside the cell?
Potentially. And what are the long-term safety implications of that, if any? That's something that probably needs more investigation.
Right. Clearance matters too.
Definitely. And then, thinking forward, now that we have this incredibly detailed blueprint of how Psi and M1-PSI create this invisibility cloak, how can we use that knowledge? Could we engineer new RNA modifications? Maybe ones that are even stealthier, or ones that have very specific tunable interactions with TLR7 or TLR8.
For different jobs.
Exactly. Imagine fine-tuning immune responses, boosting them for cancer vaccines, maybe suppressing them completely for gene therapy to avoid rejection, or perhaps modulating them very carefully for treating autoimmune diseases.
Wow. So moving from just on-off for inflammation to a whole spectrum of control.
That's the exciting frontier this research points towards, using the fundamental rules of molecular recognition to engineer better medicines.
A truly profound deep dive into the molecular secrets behind mRNA technology and some unexpected twists along the way. Thank you for joining us on this exploration of the invisibility cloak of pseudouridine.
My pleasure. Always fascinating stuff. Keep exploring and never stop asking why.